Okay, so we might begin now. Thank you. I can see a lot more people have joined. So once again, thank you all for being here. It's my pleasure to welcome you all to CureCell's 2022 annual showcase. My name is Jackie and I am the Communications and Engagements Officer here at CureCell. I'm also a PhD candidate working at the University of Adelaide, and my project involves the preclinical development of a cell therapy for type 1 diabetes. And before we begin, I want to acknowledge the traditional custodians of the country throughout Australia and their connection to land, sea and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. So today, as I mentioned, we're here for CureCell's annual showcase. And for those of you who are new to our work, CureCell is a non-for-profit organisation which aims to invest in and empower young scientists working in the cell therapy field to help them reach breakthroughs faster. And as one of those scientists, I was very excited to hear about this initiative and I'm very grateful to be involved. This is the first event that we've hosted since our official launch last November 2021. And we wanted to host this event to highlight the incredible work being done by the young scientists we have supported over the last year. And at CureCell, we believe that cell therapy has immense potential in the treatment of various autoimmune diseases and cancer. And we'll hear more about this from Professor Simon Barry, who has also joined us today, who I'll introduce later on in the session. So to get things started, I'd like to first begin by introducing our CEO, Kathy Sherrard of CureCell. So please, Kathy, give us an overview of CureCell and the work that we've been doing this year.
Thanks, Jackie. I'm Kathy. I'm CEO of CureCell. I'd also like to acknowledge that I'm speaking to you today from the traditional lands of the Kaurna people of the Adelaide Plains, and I pay respect to their spiritual relationship with their country. I also pay my respects to the cultural authority of Aboriginal people attending today from other areas of Australia. So CureCell is a tax-deductible not-for-profit addressing an anomaly in Australia's medical research environment. We and others have observed a current and growing shortfall in the pool of talent available to the medical research community. A lack of skilled, trained researchers is an identified shortcoming that CureCell is addressing. And this shortfall is of specific interest to the emerging fields of cell and gene therapies, where groundbreaking and emerging treatment developments need to be explored and understood before they can be translated into the clinic. If we expect to see new discoveries and treatments and cures in the clinic, there needs to be a commensurate number of new researchers completing their PhDs and their postdoctoral projects. And here is the anomaly. The financial support for PhD and postdoctorate researchers in Australia is currently insufficient. A typical PhD stipend stands at 2/3 of the minimum wage and is significantly less than an apprentice tradesperson wage, who of course are also paid super as well as their wage increasing per year as they progress through their apprenticeship. The stipend of $28,500 puts PhD students well below the poverty line for a single person if they have to pay for housing. It's also close to an all-time low of 30% as a proportion of average full-time earnings. Now, PhD students have already done 4 to 5 years of university studies and they've got advanced and highly valuable skill sets. They make extremely important contributions to society and the future of Australia, yet we somehow expect them to live on such a low wage for several years. And the question to be asked is how many potentially brilliant scientists don't pursue a PhD because the financial conditions they face for 3, 4 or even 5 years are just too dire. How many take 4 or 5 years to complete potentially groundbreaking research instead of 3 because they have to work part-time to fund their PhD. Now, through small and impactful scholarships, we are addressing this anomaly. We hope to provide an increasing number of scholarships that support emerging new researchers and in the process expand that personnel talent pool that is needed to underpin our emerging cell and gene therapy ecosystem. In 2022, we have supported 3 students who we will hear from shortly. In 2023, we want to support 10. So this is a bold ambition, but we hope that we can achieve it. We're going to hear from our scholarship recipients And we're also going to hear from Professor Simon Barry, who's a leading research leader in the field on what cell therapy is, the potential it holds for future treatments and even cures for cancer and autoimmune diseases, and also on why support from CureCell is necessary. Now, I want to take this opportunity to thank Jackie Scafidi, CureCell's Communications and Engagement Officer, who's pulled all of this together so brilliantly, all while she is doing her own PhD. So thank you, Jackie. And now I hope you enjoy our webinar, and thank you for joining us. And of course, if you want to find out more about CureCell, find us on LinkedIn or our website, curecell.org. And back to you, Jackie.
Thank you for that, Kathy. So yes, we appreciate that, and it's very exciting to hear this initiative is now underway. And next on the agenda, I will just take over to the next slide. So now we're going to be hearing from Professor Simon Barry, and he's going to give us an overview of a few things. But first, let me please introduce you to him. So Simon Barry is the research professor and theme leader at the Robinson Institute at the University of Adelaide. He is also the chief hospital scientist at Women's and Children's Hospital Health Network Adelaide, and is proud claim and to be a co-founder of Carina Biotech, where he is VP CAR T platform technology and gene delivery. Professor Barry has over 25 years experience in cell molecular biology with a focus on human disease. He trained in the UK and has undertaken postdoctoral training both in Australia and the US. He spent 4 years working in the US biotech sector, and this inspired his interest in translational research. He has also collaborated with national and international groups to develop cell-based therapies and technologies to enable simpler and more affordable delivery. He has patented multiple technologies and cancer targets for CAR T immunotherapy, including Carina's lead candidate LGR5, which is fast-tracked for clinical trial in 2023. In addition to cancer immunotherapies, he has a long-standing interest in applying cell molecular therapies to autoimmune diseases such as type 1 diabetes, and this includes applying CAR approaches for enhancing immunological tolerance. So I'll hand you over now, Simon.
Thank you very much, Jackie, for those kind words. I have been working in this space for over 25 years now, and my passion for immunology and immunotherapies is only really joined by the pleasure of training new scientists. And I think the mission of CureCell to make that more accessible and make that more enjoyable for our future leaders is a very worthwhile mission. And I have amongst my repertoire of brilliant scientists in training, Jackie, who's doing a PhD right now. So I thought I'd spend just a moment kind of introducing the concepts, and then you're going to hear the exciting progress that the CureCell students are making in particular aspects of immunotherapy. So I'll start from a high altitude, and then you'll come in and see the power and impact of the work that CureCell supporting. So all of this is partly to do with the immune system, and the immune system is really a complex set of cells that we all have, and their purpose is to recognize danger and fight it effectively. And that keeps us well throughout our life. And as you can imagine, there are many, many things we have to respond to. So there are lots of players, but probably one of the most important players are the white blood cells, or the T cells. And that's really where the immunotherapies that the projects are being supported to investigate are focused. So the white blood cells are very good at recognizing danger, and they can expand to fight a threat. But they can only do that when they correctly recognize that danger. So in the case of things that might affect us as humans that aren't viruses or bacteria, in the case of things like cancer, they start out from our own cells. And so the immune system has to work out that that's actually a danger threat in the case of cancer or in autoimmunity, and then fight it. And that's really where the innovation of immunotherapies has come about. So immunotherapies is a term that's used to describe any treatment that can harness and refocus the power of your own immune system. This can be drugs, it can be antibodies, and most recently, and what we're very excited about, is in cell-based therapies. And it can be applied in cancer, and it can be applied in many more emerging areas. And I think that's where the future power is going to come out. So these immunotherapies like CAR T cells, they work because they take the normal cell and then they help it to become repurposed more specifically against something like cancer. And these cells have to do that in the face of all the normal signals for the immune system saying that's something you should ignore. So it's all designed to repurpose and focus the immune response. CAR T cells got the name because they are chimeric antigen receptors. And what that really means is that the researchers that have gone before us have worked out that you can take little pieces of how immune cells function and you can connect them together. And when you connect them together, you create new potency or new recognition. And that's where the chimera term comes from. So it's a building block of parts added together to create a new functionality. And in the case of cancer, these are now engineered to specifically recognize a cancer cell and not recognize your normal tissues. And that's really been the innovative leap for CAR T therapies. You might ask, why is this a game changer? And one of the reasons it's a game changer is that in your body, you have many, many of these white blood cells, each of which might be recognizing a separate thing. And if you need millions of them to fight a cancer and you've only got thousands, you may not have a very potent immune response. If you take those cells, engineer them with your CAR and give them back, you then have millions of these cells all capable of recognizing the same threat. And that gives you a living therapy that's very specific. So this is really an exciting piece of human biology because it takes the person's own cells and rearms them. Now, this all sounds very good. You might ask the question, does it work? And there has been the most incredible revolution in immunotherapies in cancer. And it all started by trying to treat people who had leukemia. There's a particular type of leukemia, and there's a particular person you may have heard of called Emily Whitehead. At age 5, she had failed all of the available therapies and was preparing for the next step in her life journey. And she entered the first clinical trials with a CAR T for leukemia. Emily is now 10, walking around completely disease-free, and this is from a single immunotherapy. That really set the changing of the approach for how we treat cancers. Never before had there been that kind of outcome from that kind of therapy. So this has triggered an explosion in new immunotherapies to target any cancer for which you can find a specific signal. There are currently hundreds of trials going on around the world, including the one that I'm proud to be part of with Corinna. And this brings new challenges. Solid cancers are quite a bit harder to treat than leukemias because the cancer is living in the tissues and organs of the body, and your T cells have got to get there. And so this means we need to be able to build better, smarter CARs. And that's really where some of these exciting PhD projects are going in the cancer space. So it's a really important thing to acknowledge that often the engine house of new research discoveries is in PhDs and early postdoc projects, because there we can take some of the higher risks that corporate science can't take, and we can develop these new groundbreaking technologies that then will be developed into immunotherapies. And I think along with that, we're also training our future leaders. And you can't quite tell from this image, but my hair's going grey and it's time for me to retire. And I want to leave behind a legacy of bright people who bring this innovation. And I think that's the inspiring thing for me about the CureCell program. And perhaps as a final thought, what's most exciting is that we're not stopping at cancer. Now the door is open to things like type 1 diabetes or multiple sclerosis, and the emerging field of treating autoimmune diseases with CARs is very exciting. So there is a lot of work to be done, and what it needs is bright young stars who are supported by CureCell. And so I'm going to now hand over to those bright young stars, and you can hear about their projects.
Thank you so much for that, Simon. That was great. So yes, as Simon said, we are now going to hear from our bright young rising students. So this year, CureCell was lucky enough to be able to support 3 students working in the cell therapy field. Today we have Isaac Finley. He's a PhD student at the University of Newcastle. We have Hannah, who's Parry, who's also a PhD student at the Weehai Institution. We also have Lizamarie, who's an honours student here at the University of Adelaide. I won't give too much of an introduction because they will be talking about their projects themselves, but perhaps we could flick to you, Isaac, first. So we'll be hearing a short introduction from these students about what they're doing and why it's so exciting. So Isaac, I might hand over to you to begin sharing.
So can we all see those slides?
Yep.
Yep, good. Okay, so as you said, I'm Isaac Finlay. I'm a PhD student working at the University of Newcastle, and my project is on the phosphoproteogenomics of pediatric high-grade gliomas. So pediatric high-grade gliomas are tumours of the glial cell within the brain and spinal cord of children and young adults. And they encompass a broad classification of tumours, including diffuse midline gliomas, glioblastomas, and anaplastic astrocytomas. With a median overall survival of just 9 to 15 months, these tumours are the leading cause of cancer-related death in children. Current standard of care treatments include radiotherapy and chemotherapies. However, these are considered palliative, and thus there is a desperate need for novel and effective precision therapies. In some instances, pediatric high-grade glioma patients can be prescribed with precision therapies based on gene mutations harbored within their tumours. Genes are essentially the DNA blueprints responsible for constructing proteins, which acts as the functional building blocks of cells. And mutations to these genes within tumours result in changes to the structure of proteins, which leads to the uncontrolled growth of tumour cells. The study of these mutations is what we refer to as genomics and has improved our understanding of pediatric high-grade gliomas. And so, however, unfortunately, we are yet to see an increase in patient survival from sophisticated genomic studies and genomically targeted therapies. This is mainly because genomics tells us the mutation is present within a tumour, but not the effect the mutation has on the protein building blocks. Therefore, we need to analyse the proteins within a tumour to determine the effect of these mutations on tumour growth and proliferation, which we can do by investigating the abundances of proteins, known as proteomics, as well as their activity, which we call phosphoproteomics. And so this leads us to the hypothesis for our project that combining genomics, proteomics, and phosphoproteomic analysis will allow for the development of effective treatment strategies for pediatric high-grade gliomas. And so this could be through targeted therapies or through, as well covered, employing the immune system to treat the cancers. And so currently this project consists of 200 individual samples, of which we have 178 tumour samples and 22 normal controls. So these are samples of normal healthy brain tissue to be compared with tumour samples. Of the 178 tumour samples, we have 98, which are what we call tumour cell lines. So these are cells grown in the lab in flasks, and 80 are flash frozen tissue samples taken straight from biopsies and or autopsies. And so as it stands, we have performed both genomic analysis on 40 individual samples and proteomic and phosphoproteomic on 12 samples. Furthermore, of these 12 samples, we've tested drug therapies predicted to be effective on 2 pediatric high-grade glioma samples, which possess no genomically targeted therapy. These tests have been performed in vitro, so adding drugs directly to cells in plastic flasks and plates, and has provided initial evidence supporting the use of phosphoproteogenomics in treating pediatric high-grade glioma patients. For these samples, the next step is to test these drugs in vivo. So that's implanting tumour cells into mice and giving the mice our predicted therapy, which is a far better representation of how these drugs will work in patients. And so this is a rather ambitious and technical project by nature and would not be possible without funding assistance from bodies such as CureCell, as well as Run DFG, and then as well as research assistance from our team at the University of Newcastle, as well as our worldwide collaborators. So these collaborators have provided us with both individual samples to make up our list of 200, as well as technical advice on how we perform experiments. And they come from places such as Norway, Barcelona, Washington, and of course across Australia. And so with that, that's a very brief summary of the introduction of the project. But if anyone has any more questions, either during this webinar or afterwards, they are more than welcome to contact me.
Thank you so much, Isaac. That was wonderful. Yeah, I'm sure we'll definitely get some questions going in the question time. Great. So I might hand over now to Hannah to give her a brief introduction on her project. Thank you, Hannah.
Excellent. Is that all good? Excellent. So thank you so much, Jackie, and thank you for CureCell for their support and for inviting me to speak today. So my name is Hannah Barry. I'm a final year PhD student in the lab of Misty Jenkins at the Walter and Eliza Hall Institute of Medical Research in Melbourne. And as I'm from Melbourne, I'm speaking from the lands of the Wurundjeri people and would like to pay my respects to their elders, past, present, and emerging. So my PhD project involves utilising and enhancing a very important population of cells in the immune system, the T cells. So Simon went over this already, but T cells play an incredibly important role in the immune system. They help to defend the body by recognising and killing our own cells that may be infected by a virus or may be cancerous. And they do this using a receptor on the surface of their cells called a T cell receptor, which you can see in green in the cartoon here. However, in the case of cancer cells are in fact very good at evading and hiding from detection by this receptor. So there is an emerging field of immunotherapy called chimeric antigen receptor or CAR T cell immunotherapy, which redirects these T cells to be exquisitely cancer specific by genetically engineering them to express this synthetic receptor, which you can see in red in the cartoon and on the right. And this receptor recognizes proteins on the cancer cell surface that we call antigens. And as the name of the CAR suggests, it is chimeric from the Greek myth chimera, which is a hybrid made up of different animals. And this hybrid nature is very true of a CAR, which is made up of different components of different receptors to produce a signal into the T cell following cancer cell recognition. So CAR T cell immunotherapy has been incredibly successful in the clinic, particularly for patients with blood cancers such as leukemia. And the administration of the CAR T cells in the clinic involves first taking blood from the patient, isolating the patient's own T cells from the blood, engineering them to express the CAR that targets the patient's cancer, and reinfusing these T cells back into the patient. So when we talk about the success of CAR T cells in the clinic, as I said before, it's very often in the context of blood cancers. But when we try to translate this success seen in blood cancers into a solid tumor setting, for example, brain cancer, which is of particular interest to the Jenkins lab, it's often far less effective. And this is largely in part due to a problem called tumor heterogeneity, which is when the many cells of a tumor have diverse properties, such as different receptors or antigens on their cell surface. And this can make it very difficult for a therapy targeting just one antigen to entirely clear a tumor, because those tumor cells that weren't targeted by the treatment, such as the cells that you can see in purple, they will keep multiplying and outgrow. And this is a process called antigen escape. So one of the ways that we can overcome this tumor heterogeneity and antigen escape is by engineering our CAR T cells to target multiple antigens, so that we have a higher likelihood of eliminating more of the tumor. And my PhD is looking at 2 different tumor antigens to try and overcome this tumor heterogeneity. So there are many different ways that we can multi-target using CAR T cells, and I'm comparing 2 kinds. The first is on the left and involves the pooling of single specific CAR T cell populations, and this is sometimes called CAR pooling. And the second is shown on the right and involves engineering the T cells to co-express different CAR receptors simultaneously, and this is often termed a dual or bi-CAR. And I'm targeting 2 different tumor antigens. They're called HER2 and EGFR-V3, and these are often expressed in brain cancer, both pediatric and adult. So I'll just conclude a little bit by showing you some data that the dual CAR T cells are, in fact, much better at killing heterogeneous tumours. So when these pooled and dual CAR T cells were placed in a dish with tumour cells that heterogeneously express either the HER2 or EGFR V3 antigens, you can see that the dual CAR T cells in green kill the tumour cells better than the pooled CAR T cells in purple. And these both kill better than the single CAR T cells in blue and orange, which was really exciting and promising. So I'll just end by thanking everyone who was involved in my project, particularly my lab and my supervisors, Misty and Ryan, and finally my funding bodies, including CureCell, for the amazing top-up scholarship. So thank you.
Thank you so much, Hannah. That was awesome. I love the diagrams.
It's a fun cartoon.
Great. So now we'll hear from our final student, Lizamarie, who is an honours student here at the University of Adelaide. Thank you, Lizamarie.
Hi. Just 1 minute. Can everyone see that?
Yep.
Yeah, all right. So hi, my name is Lizamarie, and I'll be giving a brief overview of my findings from my honours project. So I just finished my honours project, and yeah, I'll just give a brief overview. So I looked at investigating the potential of using glypican-1 CAR T-cell immunotherapy for ovarian cancer. So ovarian cancer is the leading cause of death from gynecological cancer. Currently, the 5-year survival rate for Australian women is 43%. And most patients are diagnosed at advanced and late stages. So the most commonly diagnosed and the most lethal subtype is the high-grade serous ovarian cancer subtype, and it accounts for 70 to 74% of the cases. Current treatment strategies include debulking surgeries, as well as chemotherapeutics, and patients initially do respond to these treatments, but eventually relapse and develop chemotherapy resistance. Thus, there is a need to develop more effective treatment methods, such as CAR T-cell therapy. So I won't go in depth about this because Hannah explained it pretty well, but it's basically using your own T cells to specifically kill cancer cells. Hannah also went through this really well as well, so I'll skip this slide. So there's multiple advantages of CAR T cells. So they're highly specific for cancer cells. They're a personalized therapy because it's derived from the patient's own T cells. They have self-renewal capabilities as well, and they can generate memory T cells, preventing tumor relapse and eliminating secondary tumors. So the ideal target for CAR T cells would be a protein or an antigen that is expressed exclusively on a cancer cell or with little to no expression on normal cells. So it directly and specifically targets the cancer cells. So one potential target that I looked at was glypican-1. So glypican-1 is a protein that is expressed on the cell membrane of a cell, and it's involved in multiple cell development pathways as well. It's primarily expressed in the central nervous system and the skeletal system during embryonic development. However, it's not expressed in normal adult tissues. It's increased in multiple cancer types, including pancreatic cancer, cervical cancer, breast cancer, and ovarian cancer. And it's been investigated as a therapeutic target as well as a CAR T-cell target. However, its role in ovarian cancer has not been investigated in depth, prompting me to investigate it for my honours project. So the hypotheses that I generated from my honours project is that GPC1 is increased in high-grade serous ovarian cancer and associated with cancer progression, and that CAR T cells targeting glypican-1 are effective at killing ovarian cancer cells with high glypican-1 expression in vitro. And to test these hypotheses, the aims that I generated are to determine whether glypican-1 is increased in ovarian cancer and associated with ovarian cancer progression, and to determine whether CAR T cells targeting glypican-1 are effective at killing ovarian cancer growth in vitro. So we looked at the protein expression of glypican-1, so the protein levels within tissue area, so ovarian cancer tissues, as you can see here, and non-ovarian cancer tissues as well. So this the protein levels are dictated by the staining. So as here you can see strong brown staining indicating GPC1 positivity, and results showed that there's high GPC1 protein levels in this ovarian cancer tissue compared to that of benign tissues. We also looked at whether a GPC1 protein expression was associated with poor prognosis, and results indicated that GPC1 high glypican-1 protein expression is associated with reduced progression-free survival. These are other tumor subsection areas that showed low expression of glypican-1 within the tumor and high expression. Next, the CAR T cells were produced to determine whether CAR T cells targeting glypican-1 are effective at killing ovarian cancer growth in vitro. So this was produced using established protocols in Simon Barry's lab. I was not involved in this process, but it took 14 days to be produced. And once they produced it in their lab and then harvested it, it was provided to me for further experimental use. So I looked at whether the CAR T cell treatment affected ovarian cancer cell survival. So basically what occurred was we grew these ovarian cancer cells in plastic tissue culture plates, and then we targeted them with either glypican-1 CAR T cell treatment or untransduced T cells. So the untransduced T cells are the normal T cells from the patient that don't have the glypican-1 receptor, so the engineered receptor. And then we compared the differences between them. So this was conducted on 2 ovarian cancer cell lines, as well as 1 patient tissue. And this patient tissue is from a patient that expressed chemotherapy resistance. So results showed that once these ovarian cancer cells were treated with the glypican-1 CAR T cells, there were potent killing effects at all the concentrations we treated at. And this was even seen at the lowest concentration of 2:1, indicating the efficacy of the CAR T cells. So we also looked at a spheroid assay. So in advanced ovarian cancer patients, there's a buildup of acidic fluid in the peritoneal cavity. So that's within the tummy space. And within this fluid, there's these cell aggregates known as spheroids. And it's assumed that these spheroids are also chemotherapy resistant. So we thought it would be beneficial to see whether or not there would be there would be an effect of these CAR T cells once we treat the spheroids. So results showed that these images are 48 hours post-treatment, and within the glypican-1 CAR T cell group, there was a significant decrease in the spheroid area and spheroid growth as well, compared to the untransduced T cell group and the control media group. So this is without the treatment. This is further proving the efficacy of these CAR T cells. So a summary of my results is that glypican-1 is upregulated in the high-grade serous ovarian cancer subtype, and glypican-1 expression is associated with poor prognosis, and that the CAR T cells targeting glypican-1 reduce cell survival in monolayer and 3D spheroid assays. So the significance of my project is that glypican-1 plays an important role in ovarian cancer progression. Glypican-1 CAR T cells exhibited potent anti-tumor effects in monolayer and 3D spheroids, and overall, my project showed that glypican-1 CAR T cells have great potential to be developed as a novel immunotherapy for ovarian cancer. Thank you for listening.
Thank you so much, Lizamarie. It's I'm sure you're really happy to be done now with your honours, and you can see you've done an amazing job in such a short period of time. So well done. Just go back to sharing my screen. So now we're just going to host a very quick Q&A. I've got some pre-prepared questions here. However, if you do have any yourself that might have popped up during those presentations, please feel free to use the Q&A box. It should be down below on the bottom of your screen. So I'm going to start off. I'm sure, let me see, get everyone's face on my screen at once. Great. So I'm going to start off by asking everyone, so all the presenters today, if you were to give some advice to future young scientists looking to get into research in the cell therapy space, because we're well aware that we need more people working in this space in order to advance these therapies. What advice would you give to these students and young scientists? Not sure who'd like to go first, if there's anyone in particular.
I can answer first. So yeah, I really think getting as much experience in the field as possible. I think I probably didn't do it enough myself, and I really highly recommend it. There are some incredible opportunities for young students, both during high school and undergrad, to allow them to take placements in labs. And I think that's been incredibly valuable to decide what you want to do when you do sort of postgraduate research. And then definitely, I think doing a lot of research on the lab that you do want to join, particularly speaking as like a PhD student, and choosing labs where the people are really passionate and collegial and enjoy discussion. And there's a lot of trial and error and a lot of error sometimes that requires you to, exercise quite a lot of resilience and optimism. And it really helps to have a wonderful work environment to support you. I think, those two things really, yeah.
Yeah, I would just like to really agree with what Hannah says in terms of getting the experience you need. Like there is plenty of opportunities outside of, if say you're in your undergrad, there's like summer scholarships you can take, you can volunteer to work in labs, just get that in-lab experience, so you can do your research, find out which lab team or researchers are working in the area you want to work in, and just go up and talk to them. Researchers love talking about their research. Ask them what they're doing and see however you can help.
Yeah, great answers. I'm not sure if anyone else wants to chime in, and otherwise we do have a question that came through in the Q&A box. Is anyone else that wanted to add to that first question though, before I move on? All good. Great. So we had a question for you, Hannah. It says, great talk. And the question was, does your dual CAR T cells require the recognition of both antigens in order to activate them?
That's a fantastic question. We don't require both antigens to be present in either the sort of pooled scenario or the dual scenario. It's actually quite good because, there's no sort of molecular block to stop that from happening if one of the antigens are missing. So it's a really ideal way to sort of target heterogeneous tumors. There are definitely engineering methods where the activation of the CAR T cells in response to recognition of both antigens is incredibly useful. These are molecular techniques called sort of logic gating, in the sense that you require both antigens to cause the CAR T cell to function, and that can be incredibly powerful to prevent sort of on target off tumor effects. So you require a very specific environment for the CAR T cells to function, which can be incredibly powerful. But my CAR T cells don't look at that, but my lab do look at these other molecular techniques, which is really fascinating. Yeah.
Awesome. Actually, that's a great way to finish off that answer, because the next question that came through is on a similar wavelength, and anyone feel free to answer this one. But in regards to CAR T cell therapy, what measures can be taken to avoid potential off-target effects? For example, I'm sure people, this is the first thing that comes to mind. If there's a similar protein that's expressed on normal healthy tissue, how can a cell therapy like this avoid targeting those tissues? Does anyone want to answer that one, or Hannah again?
I could make a comment, but others would be welcome to as well. It's, the Holy Grail is a protein which is only on the cancer cell and not on any other tissue. But that's actually incredibly rare to find. So one way to deal with this is you pick a protein that is expressed at a high level on the cancer and a much lower level. And this is a dosed therapy, and you work on the principle that the CAR Ts interact strongly with the cells from the cancer that have got lots of the target, and they don't really see the healthy tissue. Now, that's something that plays out in clinical trials, is the safety profile. And sadly, often you can tolerate a little bit of side effects in your therapy if the actual benefit is strong. So it's not black and white. The other way is to design a CAR that recognizes a feature on the cell that isn't expressed, and it might be a structural change. So there's lots of really innovative approaches coming, and combinatorial approaches like the ones Hannah's developing are another tool that's in the kit. and this is actually one of the reasons why we need to support lots of innovative ideas in basic science, because it'll be the combination of those which will give the clinical benefit in the future.
Yeah, great answer. Thanks, Simon. I actually have another question came through for you, Simon, and it's a very big one. It says, will there be a cure for cancer, and is it that simple?
You know, CD19 and leukemia has cured people, so it's possible. That's what's opened the promise to all these other programs to discover new targets or to make smarter cars. Every cancer is different, and I think we're going to pick them off one by one, and we use the optimism from the CD19 cars to keep looking in other cancers. And it's just opening the door to, the more we try, the more chance there is that we will succeed. Now, you know, these therapies take time to get to clinical use. It's often 5 to 10 years, but I think the answer is that we will keep trying until these are all an inconvenience and nothing more.
Great answer. Thank you for that. I also wanted to ask a question which is applicable for everyone. So we know that Australia does really great science, but when we think of sort of leaders in the cell therapy field, I think we're a little bit further away from that. But how can Australia become leaders in the cell therapy research field, and is there an opportunity for this?
Yeah, so I might answer that. I think Australia, despite its, you know, size and sort of geography, we're quite far away from everyone else, but it has some really inspiring research and labs, and I think increased funding, more opportunities for funding and grants and stuff like that as well. So important to keep talent within Australia. And also to make Australia and Australian institutes and labs a really attractive place to draw in great international talent as well. Yeah.
Yeah, definitely. Does anyone else want to answer that one? Otherwise, we have another question that came through as well in the Q&A box. Nope. Okay, I will move on. So this is in terms of time to commercialization. How do new CAR-T approaches to cancer treatment differ compared to other new approaches that don't use cells already found in the patient's body? So I suppose small molecule drugs and things like that. This might be a good question for you, Simon.
Yeah, you know, I think this is a good example of the good that comes from bad things. I think what COVID has taught us is that you can get vaccines approved in 3 years that used to take 10. And so what that means is that what were previous barriers to the fast development and approval of therapeutics, that they're starting to be shortened. And why that matters is that, you know, cell therapies seem very complicated, but once the process is developed, they're quite quick to make. They take 9 to 15 days. And it's getting them through the trials, just like a drug or any other compound, that takes the time. So I think that the upside of things like pandemics is that we've got better at the process of getting things into trials to find out if they're safe and effective. So it will speed up.
Yeah, definitely. And another question that just came through as well. So when we talked at length about CAR T cell therapy, as it's quite hot at the moment. So what other examples of cell therapies can we think of to treat a broad variety of different diseases? So for example, Isaac, your work could really nicely link to many different approaches. Are there any in particular, other than CAR T cell therapy, that are options?
Yeah, so one of in addition to looking at the immune system, which I love, is focused on the main thing we're focusing on is looking at repurposing existing drugs. So these are basic cytotoxicity drugs. So you have a highly enriched of the specific protein, and you look to inhibit that protein. And so there are many of these inhibitors out there, but at least for DMG and other pediatric high-grade gliomas, they're so heterogeneous in terms of their genetic mutations that we don't know what these mutations do. And so you may do sequencing on one particular cancer, find out it has 5 different mutations. However, if we don't know what those mutations do, there's nothing we can do. But if we look at as I've hopefully clearly said in my presentation, if we look at the proteins to determine which is highly enriched, we work out what these mutations are. And so if we find out that this mutation is causing a high enrichment of this specific protein compared to normal tissue, so you'd want to use drugs that will mostly harm just the tumor and not the normal tissue, normal cells in the brain or in other parts of the body for other cancers. You simply want to inhibit the activity or the production of that protein, which is resulting in abhorrent tumor growth, proliferation, metastasis, and all the other deadly processes which cause the lethal nature of all cancers.
Great, yeah. It's a very exciting area, and I'm really looking forward to, I'm really grateful to be working in it myself, but also very excited for the future. We might wrap it up with one final question. This is for everyone. And we've definitely touched on it during today's event, but why is it really a good idea to invest more in the young scientists, the young and upcoming scientists going through their PhD, in particular in the cell therapy space, but in any context really? Does anyone want to start off that answer?
I guess I could start by saying it's good for me in the fact that I don't have to be bartending on weekends, which is always a good start. It's ensuring that students and young researchers have enough money to support themselves or whatnot, and to either be able to commit extra time outside of usual work hours, either on late nights or weekends, which it does happen. Research is flexible like that and works unique hours, but also in instances where maybe you don't need to work weekends and you can have that time for yourself, so that you can be rested and ready for the next week, so you can give yourself 100% to your work, as opposed to being worried about whether or not how much more hours you need to spend working at a night or at a week.
Yeah, absolutely. That's a really good point. There's nothing worse than having to work after a day working in the lab elsewhere. Ray, did you want to say something, or did you just turn off?
Hi everyone, I'm Ray Wood, the chair of CureCell. These are pretty exciting times. We're talking about a cure. 5 years ago, no one was even thinking about that. But demonstrably, as I said, there's a large number of people that are alive today that had leukemia. They've been treated with a novel new therapy, and we need to be putting some more people on the ground developing new and novel therapies. If you just think through the range and scope of the work that the 3 presenters today, Isaac talked in terms of a very early stage, amassing a huge amount of data and going through the number crunching required to actually resolve that. Hannah has come up with a novel way of expanding something. We've done it with a single CAR, but maybe a dual CAR is necessary, because sometimes the single CARs don't work. So here we've got a researcher that says, okay, I'm going to take something and build it. We're moving up the ladder. We're moving to the next step. And then you look at what Lizamarie's doing. She's saying, well, you've got these markers on the surface, but maybe there's another marker which is more specific. The range and scope that we need to look across, we need more hands at the pump making the work move forward. On behalf of the CureCell board, I'd very much like to thank Simon for your introduction and your ongoing support of what we're doing. Isaac, Lizamarie, Hannah, thank you very much for your enthusiasm. And as was one of the questions, determination and focus is not necessarily enough. It's a small contribution from someone like CureCell can actually make a difference. And so financial support is an absolute necessity to make these things work, okay? As Kathy has mentioned, we're continuously seeking donations to expand our activities. If our message strikes a chord, please visit our website, curecell.org. And please make a tax-deductible donation. No matter how big, how small, it will make a difference. Thank you very much for listening.
Thank you so much for that address, Ray. So yeah, with that, we just sort of say thank you again for attending, everyone, and thank you to the panelists for taking the time to be here, especially during your busy schedules. And we really hope that CureCell can continue to do this work in the future, and so with the hope for the generous sponsorships from future sponsors, and we can continue to do this. So thank you again, everyone, for being here. If you do, I'm just going to share this so everyone can see. If you are interested in learning more about CureCell's work, please head over to our new website that we've only recently created. So that is that should pull up now. That was curecell.org. So you can find out more about the work that we do there, and follow us on LinkedIn as well as Twitter to find out real time what we've been up to. Thank you again, everyone, for being here, and again to our panelists, and have a lovely day. We'll end it here. Thank you.